Hi, welcome to another lecture about cardiovascular system. We will look at the basic histological structures of blood, like blood vessels and heart. And let's look at the capillaries, arteries and veins. So the blood system, the blood vascular system is a closed system, of course, because we are not bleeding out every day. So inside it runs blood. It's composed of heart and blood vessels are divided into arteries, capillaries and veins, of course. We have many types of arteries, many types of veins, and also like 4 types of capillaries. And you should be able to distinguish between arteries and veins, of course, in some slides. And there are many types of capillaries. You are not really able to differentiate capillaries, only some of them maybe. Like the types of capillaries, you can just tell, okay, this is a capillary, but you should know which organs contain which capillaries, which types of capillaries. Then we have a lymphatic vascular system, We're going to talk about this system in the next lecture. There are blind capillaries starting in the tissue that runs lymph, and then they are connected to the bloodstream, but we are going to talk about this more in the next lecture. And you should know right now, you probably know that all vessels and also heart are lined by special cells, very special cells that are called endothelial cells. Endothelial cells, you know them already, endothelial cells. And they are very special. They're not just lining vessels and stuff, they have many functions also, but you should be able to describe them. They are very flat, so they look like this, very flat cells, elongated, yeah. So their orientation really is in direction of blood flow. We can see it over here. This is some cross-section of a vessel, and we can see nice endothelial cells, and you can see their orientation is in the direction of blood flow. So blood flow goes like this, so they are orientated in this way, and they are about 10 to 30 micrometers long, so they are quite long. They are thin. They contain nuclei in the center. One cell contains one nucleus, of course, in the center. And so they are a little bit higher in the center where the nucleus is. Yeah, so they are higher in the center and they are a little bit thinner on the periphery. So they have very, very long processes. They are elongated. And one would say that they are epithelial cells. Yeah, they really look like epithelial cells. And also they are sitting on a basal lamina. And you know right now that basal lamina is something that is located under epithelial cells or epithelial tissue normally. So they have its basal lamina, and also they are connected to each other with some specializations like zonula occludents or tight junction, which you already know from epithelial tissue, and also desmosomes. Yeah, so zonula occludents and desmosomes are connecting firmly together endothelial cells, and there are also gap junctions between them, so they can communicate easily. And these are very special also for epithelial cell, but please remember they are of a mesenchymal origin. Yeah, some authors call them like secondary epithelium, yeah, but they are of a mesenchymal origin. They are not exactly epithelial cells, so they are very special also because of this, yeah, mesenchymal origin. They also contain hemidesmosomes, which connect them to basal lamina, which is under epithelial cells. Okay, so they also contain hemidesmosomes. So everything is connected very firmly together, but they are also communicating with each other. That's why they have these gap junctions between them. They have basal lamina, Yeah, so when we have, for example, one capillary which is lined only with endothelial cells, they are sitting on a basal lamina that's under them. Okay, so these are very important things. And as I mentioned earlier, they have many functions. Also, endothelial cells are not just lining capillaries, they have many functions. So they are a barrier between blood and the tissue that's under the vessel, yeah, of course. So and they are a barrier, and they are also important in like providing or transferring metabolites, yeah, also between the vessel and the tissue, because you need to get some substances in the tissue from the vessels. So there is this diffusion going on in the endothelial cells, or they need to transfer the nutrients. So we can transfer nutrients by, as it's written right here, we can transfer nutrients by simple diffusion. Simple diffusion that can happen for small hydrophilic molecules, and of course oxygen or carbon dioxide, yeah, so that can happen for these very, very small molecules. Or we can transfer nutrients via pinocytosis, which is an active transport of glucose, amino acids, electrolytes. And we have these pinocytic vesicles inside them. We can see pinocytic vesicles in the electron microscope, and they are around 70 nanometers big. Yeah, so pinocytosis, 70 nanometers big vesicles. And these pinocytic vesicles are transported over the cell. Yeah, so they are transported through the cell to the basement membrane that is under the endothelial cell. This is called transcellular pathway when they are transferred through the cell. Or this transfer can happen also between endothelial cells. Yeah, so on the lateral side of the cells, between them, and that's called paracellular transport. Yeah, para means somewhere like in not like in between, but next to something. Yeah, so transcellularly or paracellularly it can happen, this pinocytosis. So and here we can see pinocytic vesicles over here. So probably this is some kind of capillary over here. This is a lumen of capillary, and this is a process of endothelial cell over here. So we are not able to see the nucleus of endothelial cell, only just a process of endothelial cell. And these circles over here are pinocytic vesicles. So these are pinocytic vesicles. They are about 70 nanometers big, and they are transferring metabolites from the lumen, from the capillary to the tissue that it's around, of course. You're probably not able to see these, me neither, but there should be a basal lamina under endothelial cell over here, but we cannot see it very well. There should be basal lamina. And what do we have here next? We have probably a cardiomyocyte over here, cardiomyocyte. And how can I tell this? I see myofibrils over here. So this is one myofibril, this is the second one over here. So this is muscle tissue, of course. And when I see myofibrils, I have two choices. It's a cardiomyocyte or a muscle fiber. Yeah, so I have two options. But I see probably very big mitochondria over here with a lot of crysts inside, many of them. So probably this is a very, very active mitochondria. And I think it's a cardiomyocyte. Yeah, During oral exam, if you would say it's a muscle fiber, it will be very much okay. We will discuss this situation and we will ask you what could it be more than a muscle fiber. Yeah, and the correct answer is cardiomyocyte, of course, so don't worry. It's not very easy to recognize it's a cardiomyocyte actually, but we have a membrane that is of course around the cardiomyocyte, so it has its membrane, and also cardiomyocyte, and once again we are not able to see it right here, but should be enveloped by something called superficial lamina that is all around the cardiomyocyte. So we have endothelial cell over here, the process with pinocytic vesicles. I can tell it's an endothelial cell because it's very typical that we have just this long process that containing many pinocytic vesicles. That's very, very common for endothelial cell. And here is lumen, and probably in some pictures you will be also be able to see erythrocytes over here, like very electron dense structures. So that could tell you it's a blood vessel. And underneath the endothelial cells there is a basal lamina, and also we can see a superficial lamina over here. So this diagram shows transport across endothelial cells. Note these hemidesmosomes, which are connecting individual endothelial cells to the basal lamina, that's under endothelial cells, of course. And we have also desmosomes, which are connecting two endothelial cells together, also with zonula occludents. This is called like the junctional complex. Don't forget about the gap junctions that are also somewhere in between. Yeah. So we can see the pinocytosis over here. So there are many pinocytic vesicles, and these pinocytic vesicles can go transcellularly to the other side where the basal lamina is, or they can go to the lateral side, and that's like the transport paracellularly. Yeah, so when they go between the endothelial cells. Also, what's very interesting here are these orange bodies over here. They're not really orange, but for the purpose of this picture, also the mitochondria are green, so that's not really happening, but you know that. And so these orange bodies over here, you might heard of them, but you will hear more about them in your next years probably, because these bodies are called Weibel Palade body, and they contain von Willebrand factor. Von Willebrand factor, which is a glycoprotein, is a hemocoagulating factor, and it's binding to factor 8. Yeah, so we need this factor for coagulation, and there are certain mutations. There might be certain mutation in this factor which cause the patient to bleed very easily. So you should know that granules in endothelial cells contain these Palade bodies. And also certain drugs that are used to treat patients can release this body, these granules from endothelial cells. So we can higher the number of coagulation factors, the von Willebrand factor, when we administer some drugs to the patient. Yeah, for example, desmopressin is one of the drugs that can higher the levels of von Willebrand factor. And also this Weibel Palade body contains also the factor 8. Factor 8, which is also in this coagulation cascade, is very important. Yeah, so you should know this. We have also a picture from electron microscope, these Weibel Palade granules or the bodies. This is how they looks like. You probably, there's no need for you to recognize them during oral exam. It's just for you to imagine how they look like, really. They are very similar. I think they look like a little bit like mitochondria, but Yeah, I probably wouldn't be able to recognize them, and that's pretty normal. Here we can see the pinocytic vesicles over here. Yeah, pinocytic vesicles right here. Okay, we also recognize the endocytosis that is clathrin dependent, so like receptor mediated endocytosis. So as you remember probably from cytology, we have many ways how to transport substances between cells. There was this pinocytosis, which wasn't receptor mediated, but we have also pinocytosis which is receptor mediated. And when we have a receptor that binds some kind of substance, we need a clathrin. We need a clathrin as a protein which polymerizes under the biological membrane of a cell and envelopes the substance. Yeah, so it's called receptor-mediated endocytosis. We have something called coated pit, and this coated pit, coated pit, it's turning slowly into the coated vesicle. That's normal pinocytic vesicle, but it's coated because there is clathrin all around this pinocytic vesicle. Okay, so this is not a very good picture, but I think you can imagine how it looks like. In electron microscopy, it will be more dense on the periphery. Yeah, so you can recognize these vesicles. They look really the same as normal pinocytic vesicles, but they are a little bit more dense on the periphery, so nothing special about it. So, these clathrin dependent or this endocytosis is transferring special molecules like, for example, cholesterol, transferrin, and lipoproteins. These are very important because we differentiate 5 groups of particles that contain lipoproteins, like LDL, which is a low density lipoprotein, a very low density lipoprotein, high density lipoprotein, and chylomicrons and other stuff like this. But you're going to learn about them in biochemistry. But right now you should know at least about LDL, which is referred to as a bad cholesterol. It's not very bad because normally you need cholesterol in your cells, but when you have higher levels of cholesterol, it's bad. Yeah, so that's why it's called bad cholesterol, because it's transferring cholesterol into the cells. And LDL, it's normally in the blood in some levels. It's a lipoprotein. It's about 19 to 22 nanometers big. It's not very important, but endothelial cells have receptor for these LDL particles, and they are oxidized there. They are oxidized, yeah, because normal cells produce free radicals, and these lipoproteins are oxidized there, and that is very important for the formation of plaque, atherosclerosis, and stuff going on. I will get to atherosclerosis at the end of my presentation. So, but right now, just remember there are many lipoproteins in the blood. They contain cholesterol. One of the particles is called LDL, low density lipoprotein, and it's normal to transfer cholesterol to the cells because they need cholesterol. So let's go somewhere else right now. So here is a summary of the function of endothelial cells. So it's not just like the barrier. It's not, endothelial cell isn't there just for the transfer or transport of nutrients and for creation of barrier. It's also important for the modulation of blood flow, yeah, and vascular resistance. It's releasing and producing and releasing many factors as vasoconstrictors and vasodilators. Yeah. Also, it's producing some growth factors. It regulates immune response. Also, endothelial cells are producing extracellular matrix. Yeah, that's very important because they contain basal lamina. And what is going to produce basal lamina underneath them? They're going to produce basal lamina. And also they inactivate many substances like bradykinin, serotonin, thrombin, which are very important substances in bloodstream. They do many things. And also they are involved in lipoprotein metabolism, yeah, and production of free radicals. And also the maintenance of nonthrombogenic barrier is also very important. So, and once again, we are getting to this barrier because probably it's the most important function, yeah, to provide a barrier. And we can find very important barriers in typical environments, for example, in a respiratory system, yeah, between the alveoli, which contain gas, and the blood, of course, because we need to transfer blood gases like oxygen. We need to get oxygen from our lungs, so there we have a very, very good hemorespiratory barrier. And also in placenta, for example, placental barrier. And you heard about placental barrier earlier in embryology lectures. So here we have a barrier between the blood of the fetus and the blood of the mother, because blood of the mother and blood of the fetus are not mixing together. There is need for some kind of barrier between these bloods. So, and also we have another barrier which is not mentioned here, but also in the kidney. I'm just going to write the kidney. We have another barrier, like the hemourinary barrier, because we are producing urine from the blood, actually. From the filtration of the blood, we are producing urine, so that's why we need some kind of barrier also in the kidneys. So here we have a nice picture. This picture is on oral exam for sure, so remember this picture is very important. Here we have a process of endothelial cell over here. So once again, we are not able to see a nucleus or something, just a very thin process of endothelial cell. So this is probably a capillary over here. Once again, we are not able to see erythrocytes inside, only just some substance here, plasma or blood or whatever. But we are not able to see erythrocytes, doesn't matter for us. But we know this is endothelial cell. And why do we know it's an endothelial cell? Once again, it contains a lot of pinocytic vesicles, a lot of them. Yeah, so many pinocytic vesicles. Some of them are more electron dense on the periphery, so probably this is some like clathrin mediated vesicle, or over here receptor mediated endocytosis. Some of them are not so electron dense, so probably it's normal pinocytic vesicle. We cannot tell for sure, yeah, but we can see many pinocytic vesicles, so it's an endothelial cell. And here there is some other process of another cell which is also similar to endothelial cell because it's very thin. We can only see its process, and that is a pneumocyte type 1. Pneumocyte type 1. And when we have type 1, we probably also have type 2, but we are going to talk about it more in the respiratory system. So this is a pneumocyte type 1, which is a very thin cell. It's a very special cell that's located in the lungs, in alveoli. And between this pneumocyte here and endothelial cell, there is another layer over here. And can you guess what is it here in between? That's probably just some extracellular matrix over here, yes? But remember, endothelial cells have basal lamina. And also pneumocytes type 1 have basal lamina. So this layer over here, these are the fused basal laminae together. Two basal laminae together are fused, so they create a layer between these two cells over here. Yeah, so this is just this wave over here. They are fused basal laminae over here. And can you guess what's here, inside here, this white space? There's just gas, okay, air, just normal air in alveoli. So we have air-blood barrier when we transfer gases, blood gases between over here by a simple diffusion, okay? But there are also pinocytic vesicles which are not transferring gases but other substances that need to be transferred also. So, okay, this is normal air-blood barrier. This is a picture from placental barrier over here. So these are fetal erythrocytes over here. These are fetal erythrocytes. So we are somewhere in a villus, in a small tertiary villus over here, this whole picture. And as you know, the tertiary villus or normal villi of a placenta are covered with trophoblast. So they are covered with trophoblast, and trophoblast is made of two layers. It's made of syncytial trophoblast. This is a syncytial trophoblast over here. It's very thin. And you can see syncytial trophoblast contains also microvilli on the surface. They're not many, but they are. They are helping with absorption of stuff. Yeah. So these are the microvilli. This is the syncytial trophoblast, and underneath the syncytial trophoblast we can sometimes find very light separated cells of cytotrophoblast. This is a cell of cytotrophoblast, and over here. Yeah, so we can see it nicely on the picture from electron microscope. We will be able to see this also in light microscopy. But what's important is that this is some kind of vessel containing blood of the fetus. We can see erythrocytes here, but also we are able to see erythrocyte over here on the other side. And this is erythrocyte that belongs to the mother. Yeah, and you can see these bloods are mixing together, but there is some kind of barrier which is made of syncytial trophoblast, which is sitting on the basal lamina, and also endothelial cells which are lining this vessel over here and its basal lamina. And we will get to this once again during female reproductive system, so don't worry. Just note that there is something called placental barrier. So now let's get to the capillaries, finally. We describe the capillaries first of the vessels. They are the smallest, of course, and they are composed of one layer, the single layer of endothelial cells, and basement membrane that is under endothelial cells. Yeah, just remember, basement membrane is basal lamina that you already know, which is made of lamina lucida and lamina densa. So basal lamina plus reticular fibers. Reticular fibers. So when we have some capillaries that are normally in the tissue, of course they're going to have reticular fibers around them. So we not say that there is a basal lamina underneath them. We say that we have basement membrane. When these capillaries are in the tissue, we have basement membrane, and that's just the basal lamina with reticular fibers, and we can see basement membranes in a light microscopy. We can describe them in light microscopy. So please don't describe a basal lamina in light microscopy, because actually it's so thin you're not able to see basal lamina. Only in electron microscopy you can see basal lamina. So they are about 4 to 9 or micrometers in diameter. Sometimes they are also bigger in diameter. In special sinusoids, they can be a lot of bigger, but this is a normal diameter of capillaries. In length, they can be 1 millimeter long, but even more. Yeah, capillaries can measure several centimeters, so they are about this long. Okay, we can find them in kidneys, adrenal glands. They are longer there, of course. In the kidneys they are longer, and this is some kind of summary. Yeah, they are about 50,000 miles together in length, so that's quite strange to imagine. And also some capillaries have pericytes around them. Not all of them, but some of them have pericytes in the wall below endothelial cell. Yeah? So the normal composition of capillaries are one layer of endothelial cells and basement membrane. Yeah? But sometimes we can find also pericytes, which are another cells around under the basement membranes of capillaries. They contain actin and myosin filaments in the processes, so pericytes are able to contract normally. So here we have just some kind of scheme, and the larger vessels from the capillaries are arterioles. So these are larger vessels. We're going to talk about them. They have layers of smooth muscle cells around them, and they are branching and branching. And here we have precapillaries over here, precapillaries, which are about 40 micrometers in diameter. They contain some smooth muscle cells, and they are also branching and branching, and they create capillaries. So normal capillaries over here. Yeah, so this is a capillary and this one is a capillary. And these capillaries are lined sometimes with pericytes. Yeah, so in bigger vessels we have normal smooth muscle cells around, but in smaller vessels like capillaries we don't have smooth muscle cells. That's very important. We don't have smooth muscle cells. We just have these pericytes, which are lonely, sometimes sitting on the capillaries, causing the contraction to facilitate blood flow through capillaries. Then these capillaries change into veins, the venous system over here. So we have post-capillary venules over here, post-capillary venules, and also bigger venules over here, and blood flows back to the heart, which is where is deoxygenated blood goes back to the heart. Yeah, and we are going to also differentiate these vessels, bigger vessels over here. But just have in mind the schema. We have like normal arteries here. There is just some kind of capillary system, which is very important, capillary system, and then everything goes back to the venules over here. So capillaries can be divided into several groups. We just don't have one capillary and that's all. That would be very nice, but it's not like that. We have many types, and we divide capillaries into two basic categories, and that is typical capillaries, that are typical capillaries over here, which contain continuous basal lamina underneath. So typical capillaries, continuous basal lamina, and also atypical capillaries, which don't contain continuous basal lamina, and this is true for very special capillaries called sinusoids. And it's typical they have discontinuous basal lamina under the endothelial cells. So typical and atypical capillaries. The first group of typical capillaries are continuous or somatic capillaries, continuous. We find them mainly in muscles, in the CNS, that's very important, CNS, muscles, the thymus, lungs, exocrine glands, and in normal connective tissue. So the lining is continuous. Endothelial cells don't contain any pores, any fenestrations. The lining is very, very continuous. There is no connection, like the substances need to go through endothelial cells. It's not easy to transport substances, so this barrier is very important for the CNS, very important. But it's also in other types of tissue, yeah, as you can see also in muscles, lungs and other connective tissue. It's very important also in the thymus to have continuous type of capillary. Another type is visceral or fenestrated type of capillary. So we have fenestrated type of capillaries, and they have fenestrations inside them. And how can we describe fenestrations? Fenestrations are just holes inside the endothelial cells, so they are holes inside. But what's important, there is a diaphragm in the holes of endothelial cells. So there is a thin layer that's covering the hole. Yeah, it's not just like the space you can get through very easily. There is some kind of membrane, the diaphragm, which is very similar to normal biological membrane, but it's thinner than a normal biological membrane. So these are the fenestration. Fenestrated capillaries are important for the endocrine glands. So you can transfer hormones easily, so endocrine glands, they can be found in digestive system. Peritubular capillaries in the kidney, of course, yeah, so you should remember at least these three. So when we are going to describe endocrine glands, like for example hypophyses or adrenal gland and stuff like that, you should know from now on they contain fenestrated capillaries, because you need to transfer hormones very easily. Okay, the third category is capillaries with pores, and also pores are very similar to fenestrations. Fenestration is almost the same, but there is one difference, and that is that these fenestrations don't contain diaphragm inside them, so that's why they are called pores. There are really holes inside endothelial cell, and this is sometimes maybe a little bit confusing because there's the capillaries with pores. So some of you sometimes describe it, and also that was my case. I thought earlier that there are pores between endothelial cells. So there is one endothelial cell and another one, and there is a hole between that is called a pore or a fenestration, or they are continuous, so there is nothing between them. But true is the opposite, because we have endothelial cell like this with the nucleus, and the pores are really inside the endothelial cell like this. So we don't have pores between endothelial cell, we have inside them. And it's sometimes confusing because when you see a picture from electron microscope, I'm going to show you, there is just a section from electron microscope. It's just a section. So it sometimes looks like these pores are between two endothelial cells, but it's not true. Yeah. You should remember from now on, capillaries with pores that are fenestrations without diaphragm are only in the kidney. So they can be found only in the kidney, in the structure called glomerulus. So we have something called glomerulus. There's just a bundle of capillaries. It's a glomerulus, and only in glomerulus we have capillaries with pores. Nowhere else. Okay? So you should remember this right now. It's very important. So, and then we have atypical capillaries, which are blood sinusoids, and they have discontinuous basal lamina. That's very important. It's a typical feature, and they can contain also fenestrations, so we can find fenestrations inside them, and sometimes there could be a diaphragm in this hole, but sometimes we can find also pores there, but it's not typical. Yeah, these capillaries, these sinusoids are very big. They are bigger than normal capillaries, and we can find them in some tissues, like in the liver, a bone marrow, and the adrenal gland, sometimes the cortex of adrenal gland, and also in adenohypophysis. So as I told you before, the hypophysis contains these fenestrated capillaries, because hypophysis is an endocrine gland, but sometimes endocrine glands also contain sinusoids. This might be a little bit confusing for you right now because you don't know the organs, but when you're going to learn about organs, focus on the vessels sometimes, please, because we're going to ask you, are these vessels containing fenestrations, or can we find pores here? Can you find sinusoids here in this tissue, or what are the basic type of capillaries here? So this is very important stuff, and you should know which type of capillaries you have in a specific tissue, because it's very important for the transport of nutrients and hormones that are produced there. So please learn about it more and study it more. So the first type is continuous somatic capillary over here, and this is a typical capillary, really very typical capillary. There are no fenestrations in endothelial cells, so there is a lumen of endothelial cell. We can of capillary. I'm sorry. This is a lumen of capillary. This is our capillary over here, which is lined with basal lamina underneath, and we can see two endothelial cells over here, or probably even more, because what we can see are these connections between them. The arrow is pointing into the connections, which contain tight junctions, the desmosomes, zonulae, occludentes, and also gap junctions, like the next side, the nexuses over here. And these are the thin processes of endothelial cells over here. There is also some kind of connection, so probably we are able to see 3 endothelial cells lining the capillary. And we can see also, probably more on this picture, that there are many pinocytic vesicles inside. That's a typical feature for endothelial cell, and we can see these pinocytic vesicles over here, right here. So there are no fenestrations. Everything is connected tightly together, and this is important for the central nervous system to have very tight barrier inside. So they are a component of blood-brain barrier, and also for the thymus, it's very important. Blood-thymus barrier right here. Probably what we can see also here in this picture is a cell called pericyte. As I told you before, pericyte is a cell that sometimes lines the capillaries, that contains contractile elements like myosin and actin, so it can contract and facilitate blood flow. So this is another picture of somatic capillary, which is lined only by 1 endothelial cells over here. So once again we can see a nice nucleus here inside, and very thin processes that are lining a capillary. Once again, somatic capillary over here. Next we have fenestrated or visceral capillaries. They are found once again in endocrine glands, and at site of fluid and metabolite absorption. Yeah, so it's typical for gastrointestinal tract. and also peritubular capillaries in kidneys. Peritubular means that there are some capillaries around tubules of the kidneys which are absorbing stuff. So here we have a nice red blood cell inside. There are probably two, and this section looks very strange, but okay, that sometimes can happen. This is not normal structure of red blood cell, but there are probably two of them somehow close together, so that can happen. And what we can see right here, there is only thin and process of endothelial cell, and we can imagine there are some kind of like holes inside the processes of endothelial cells, which are lined with diaphragm. We can probably see it better on this schema over here. So this is very small magnification to actually see the fenestrations over here, but the fenestrations are small, like small holes which contain the diaphragm. Yeah. These are here, they are called like the very thin are perforated by pores. Okay, but these pores contain diaphragms, so they are called fenestrations. Yeah, so just remember the name fenestrations, and from now on, when there is a diaphragm, called it like fenestrated capillaries. Okay, fenestrated capillaries. We can see it right here very clearly. So this is probably, this together is probably just one process of endothelial cell over here. And we can find pores which are lined with diaphragm, so they are called fenestrations between here in this lateral process, not between lateral processes, but in the lateral processes. And we are at the third type of capillaries, and these are capillaries with pores. So pores don't contain diaphragm, and these capillaries which contain only pores inside them, no fenestrations, and yeah, only pores without diaphragm, can be found only in the kidney, in the glomerulus, at the site of hemourinary barrier. So remember this, hemourinary barrier. So we are definitely in the kidney in this picture, because we can see a capillary over here. There's a capillary, and we can see these things over here. Okay, these parts, like they were separated, some structures over here, but really what's going on, this is probably a process of one endothelial cell which is perforated. And once again, when I'm going to draw you like endothelial cell, how it looks like, and here we have many pores inside the process, and when you do the cross section somehow like this, you will get like separated cytoplasm, and you get the pores between the parts of cytoplasm over here. So this thing over here, this is a pore, and also this is a pore, and you can see it clearly, very well. You can see it very well right now that we are not able to see fenestrations. There is no fenestration. That's why we call it a pore. So this is a capillary which contains pores. Here we have some layer over here, which you can probably read is a basement membrane. So there are fused membranes over here. In the kidney it's called the basement membrane, glomerular basement membrane, because we are in the glomerulus. Right here we're going to learn more about it, don't worry. And on the other side we have once again some cells, some parts of cells, and these are the secondary processes of very special cells that can be found in the kidney called podocytes. Podocytes are very big cells, as you can see right here, but they have primary processes and secondary processes, and secondary processes are called pedicles. Which are sitting on the capillaries, and they are creating a filtration barrier between blood and the urine, because we have urinary space right here, which is containing and which will contain urine after the filtration from the blood. And these pedicles contain diaphragm between them. So we can find diaphragm in this picture, but not in the endothelial cells, not in the pores. We can find it between the secondary processes of podocytes, which are sitting here on the capillaries and providing the filtration barrier over here. But we're going to learn more about this in urinary system. Once again, in higher magnification, here is the capillary, and I'm sure this is a capillary. I'm not able to see many pinocytic vesicles here, so I'm not sure it's an endothelial cell, but I can see a very thin process over here that contain pores. Yeah, very thin process that contains pores right here. So I'm sure this is a capillary with pores, and the blood that's inside here will filtrate like this, in this way. So also basement membrane here is very important filtration barrier, and we will have urine in this space over here. So, and the last capillaries are the sinusoids, which are not normal. As I told you before, normal capillary is like 4 to 10 micrometers in diameter, but sinusoids are bigger, a lot bigger. They are 30 to 40 micrometers wide, and they contain fenestrations. Sometimes we can find also pores inside them. So as you can see it right now, there are probably these are the pores, but they can contain this diaphragm between them, like here inside. So these are called sometimes fenestrations. The basal lamina is discontinuous, it's not normal. So these sinusoids, these capillaries, are specialized for the transport of some bigger substances. That's why we can find them in, for example, in bone marrow, red bone marrow. Because what's happening in the bone marrow? Hemopoiesis, yeah? So you're creating cells, the whole cells that needs to go to your peripheral blood somehow. So that's why you have sinusoids inside the bone marrow, because you create cells and you need to transport those cells in the peripheral blood. So you need capillaries that are very discontinuous, because in that case, erythrocytes, here we have inside, but erythrocytes can move from this space around. They're developing in the bone marrow, so they can move inside the sinusoid. Okay, so that's why we have these sinusoids inside bone marrow. And these fenestrations are very wide. These fenestrations are about 0.5 to 3 micrometers wide, so they are bigger. So really the erythrocytes can squeeze between these fenestrations and transport to the sinusoid. So this is how it looks like. So here we have a nice picture from a bone marrow, and this is very wide sinusoid over here. So you probably wouldn't be able to tell that this is a capillary, because it's very, very wide. It's very wide. It almost looks like some kind of venule or something like that. But you should know that bone marrow contains sinusoids, which are very wide, and this is a pretty normal picture. Yeah, so this is a sinusoid, the longitudinal section over here, and we can see erythrocytes. And also what we can see in bone marrow, here we have nice developing cells over here. So they are dividing and developing, and here we have adipocytes. So there are many adipocytes. They are univascular, and we have many of them in the bone marrow. And here we can see nice fenestrations in the wall of sinusoids over here. So what do we have next? Is a picture from a liver, because also in liver we have sinusoids. Yeah, so here we can see nice endothelial cell which is sitting here. It's lining sinusoids here in the liver. We can see right here that is wider where the nucleus is, so it's wider over here. But here we can see nice small thin process that is protruding from the center of the endothelial cell. And of course there should be a basal lamina over here, but we cannot see it clearly in this magnification. And here we have nice erythrocytes over here. But what we can see on the other side, these are hepatocytes over here. So these cells are hepatocytes. I'm going to write it down. Hepatocytes, which are normal cells of the liver. You can see they do contain microvilli over here, microvilli, and there is some space between hepatocytes over here and endothelial cells, and this space is called space of Disse. Space of Disse. And you're able to find this space of Disse also in light microscopy. You can see there is some kind of space between endothelial cells and hepatocytes. So in practical trainings, during practical trainings, we're going to have a look on the liver, and I can show you space of Disse, or somebody else will show you, someone else, I'm sorry, but you will see it. Okay, so there is some kind of space between. So, well, we discussed the capillaries finally. That was very important topic. So remember the basic types and where can we find them. But we also have some basic layers of the wall in vessels. So capillaries were just like endothelial cells with basal lamina around them, sometimes pericytes, and there was no need for other stuff that was around. But when we have bigger vessels, we differentiate three basic layers of vessels. So from now on, we're going to differentiate a tunica intima, or sometimes called interna. That's okay. Sometimes you can find this other name, but in our department we are like the intima more, the tunica intima. It's an inner layer of the vessel. It's made up of endothelial cells, of course, because they line vessels, and subendothelial connective tissue. There is some kind of layer of connective tissue underneath endothelial cell. So, okay? So, endothelial cells with subendothelial connective tissue. Then we have tunica media, which is a middle layer. It's the thickest in arteries. It contains a lot of smooth muscle cells. So you can find smooth muscle cells in the tunica media. Yeah, but it really depends on if we're looking in the arteries. If we're looking on the arteries, there are many smooth muscle cells inside, and it's the thickest layer. But when we have a tunica media in the veins, it's not so thick, it's thinner, and it doesn't contain many smooth muscle cells, so it really depends. But now on, just remember it's a middle layer, it's thickest in arteries, and it contains a lot of smooth muscle cells. But it also contains elastic fibers, so it contains elastic fibers, and it also contains reticular fibers. Not so many, but it contains them. Okay, so it's not just muscle cells together, it's also elastic fibers, reticular fibers, and other stuff. Then we have tunica adventitia, or tunica externa, and it's the outermost layer of the vessels, and it's the thickest in veins. So remember, the thickest layer in veins is tunica adventitia, and the thickest layer in arteries is tunica media, and that's how you can differentiate veins and arteries from each other. Yeah, because the thickness of tunics that are here. And also we have something called vasa vasorum, or vessels of the vessels, because when the vessels are very, very big and thick, you know that they are supplied by nutrients from diffusion of the blood that is inside the vessels. So there is a diffusion of substances from the blood that is inside, but adventitia, it's the outermost layer which is not supplied very well from the diffusion because the path is very long. So we can find vasa vasorum in adventitia, which supply the adventitia and also part of tunica media that's there. So we will be able to find something like this. Also, what is very important in layers, it's their orientation. Because of course we have many sections of vessels. Sometimes we dissect the vessel more longitudinally, sometimes there is a cross section, and you must differentiate the section. Is it the cross section, it's the longitudinal section, or what's going on? So I'm going to draw it for you. So here we have a nice vessel over here. So this is a vessel lined with endothelial cells, and of course we have this tunica intima, and you know that endothelial cells are oriented longitudinally in the way of blood flow. So tunica intima and also the fibers that are in the subendothelial connective tissue are oriented longitudinally. So it looks something like this. Yeah, longitudinally, like this. So this is a tunica intima. Then we have a tunica media, which contains smooth muscle cells and elastic fibers and reticular fibers, and this layer is oriented circularly. So these elements that can be found in tunica media are in circle all around like this. It's very important, yes. And in tunica adventitia we can find also smooth muscle cells. There are many muscle cells in veins in tunica adventitia, not so many in arteries, but in veins we can find many smooth muscle cells also here in tunica adventitia. And these muscle cells and also elastic fibers that are also here, plus reticular fibers that can be found here, and smooth muscle cells are also oriented longitudinally as tunica intima, like this. So this is the basic orientation of layers. You should know this and you should describe this. And based on that, if we are in the artery or vein, so we can tell, okay, so probably this is vein because in adventitia I can see many smooth muscle cells and it's the thickest layer. And in the arteries, the thickest layer is tunica media, which contains many smooth muscle cells. So let's move on right now. We also classify vessels according to diameter. So we can find very large arteries that are over 1 centimeter thick, so very thick, large arteries. These arteries are of an elastic type. We're going to talk about them. It's, for example, aorta and her branches, and also the pulmonary trunk. Okay, so remember these, very important, elastic types. Then we have medium or middle-sized arteries, which are about 2 to 10 millimeters in diameter. Then we have small arteries, and arterioles are smaller than 0.5 millimeters. These large arteries are often elastic type. They need to be very elastic, and medium-sized and small arteries are of a muscle type, because they do not contain that much elastic fibers, so we are not calling them elastic, but they contain a lot of muscle cells, smooth muscle cells, so they are of a muscle type. Okay, then we have a precapillaries. We talked about them, not so important right now for you. Then we have capillaries. These are important, you should know the basic types. Then we have post-capillary venules. Collecting venules, muscular venules. Okay, you should know that we have some collecting venules and muscular venules, but what is important for you is to differentiate venules. Okay, so differentiate capillaries and venules. Then we have a small and medium vessels, and the last one are the largest vessels that are in our body. So once again, 1 centimeter and larger, like the vena cava, superior and inferior, and that's, I think, the largest vessels that can be found in the human body. So we're going to have a look on each vessels right here. So let's start with arteries of elastic type, so the biggest arteries that we have in our human body. This is an aorta over here, aorta, but it's hard to differentiate. This is really aorta. It could be some branches of aorta, the biggest branches, or it could be trunk, the pulmonary trunk. So we just know this is an aorta. But what we can tell based on this picture is that that's an artery of elastic type, and is one of the biggest arteries here, and of course we can tell it's an artery. And how can we tell that? So once again, we have these 3 basic layers. You should be able to describe these layers, because we have many specimens, the many slides that contain vessels, and on each slide you should be able to differentiate vessels and describe basic layers. They're asking this often during oral examination. Yeah, so probably you have a nice slide, for example, I don't know, like the liver over there, and there will be some very big vessel inside the liver, and they will ask you like, what's this? It's a vein, it's artery, and how can you tell that? So this is an artery. We can see a nice layer of, maybe it's labeled over here, yes, it's a nice layer of tunica intima over here, which is made of endothelial cells that are here on the top, endothelial cells, and subendothelial tissue over here. Just normal connective tissue, loose connective tissue under endothelial cells. So we can find normal, like also collagen fibers, elastic fibers, reticular fibers, very thin fibers over here. And what we can see next is a tunica media. It's labeled with B, so this is the tunica media, which is oriented circularly. So this is probably the cross section. This is the cross section because we can see its circular orientation right here. So this is tunica media, and it's composed of smooth muscle cells. So we can find smooth muscle cells here. And also there are many elastic fibers over here, and that's why it's called an elastic type artery, because there are many elastic fibers, and not just elastic fibers, they are connecting and fusing together, and they are creating whole membranes. So there are elastic membranes here. We're going to see that on the next picture. It's probably more visible for you to see those elastic membranes, but remember there are many elastic fibers here. And these elastic membranes contain many holes inside them. So that's why they are called fenestrated membranes, because they are like perforated. They're not just like the layer, like the real membrane, but there is a membrane that has many holes inside, and that's why it's called fenestrated membrane. And the last tunica over here is a tunica adventitia. Which contains, it's made of loose connective tissue. Normally it contains also elastic fibers, reticular fibers, and it contains some smooth muscle cells, it could be. And we can see that tunica adventitia over here contains vessels. another vessel, so a vessel that contains a vessel, and it's a vas, vasorum, or vessels of the vessels, which these vessels supply adventitia and also part of tunica media over here. And the other layers are supplied from the blood that is normally in the vessel, so tunica intima, and the other half of tunica media is supplied by the normal blood in the lumen of aorta over here. So here we have very special staining. This is a staining with orsein. You should know this because we have also many slides stained with orsein. Not so many, but just like 2 of our slides are stained with orsein, which is a special staining for elastic fibers. So that's how we can visualize elastin in aorta, which is an artery of elastic type. So what we can see here, it's okay, of course, many layers. We are not able to differentiate many things here because there are also smooth muscle cells here inside the tunica media, but we are not able to see them very properly. But what we can see are these lines that are curved a little bit, like wavy lines over here, and these are the fenestrated elastic membranes. There are many of them inside the aorta, many of them, and with age the number of elastic membrane increase. So with age the number of elastic membrane increase. The older adult has like 60 to 70 membranes in aorta. Yeah, so really the smooth muscle cells are stuffed between elastic membranes which have fenestrations inside them, so between there are many smooth muscle cells. We can also see it in this picture over here. We can see smooth muscle cells very properly. This is the nucleus. This is a cigar-shaped nucleus of smooth muscle cell over here and also here, but you can see membranes, the fenestrated membranes, very nicely. And the orsein over here is staining them in brown color. So orsein should be like more brown color, but I think this looks kind of pink, but never mind. Yeah, so orsein is blue color or something between not blue, the brown color. Sorry, brown color or something between brown and orange. So, but we can see the membranes very well. Why do we increase in elastic fibers? Because there is some compensation going on to pulse wave. Because imagine if the aorta wasn't elastic enough, the pulse wave from the heart, the beat from the heart, will cause largening and widening of aorta. Yeah, so the aorta needs to be elastic. These are the fenestrated membranes. So now you can see nice picture how it really looks like. We're not able to differentiate, or there are no smooth muscle cells right here in this picture. We can only see fenestrated membranes over here. This is how it looks like. Yeah, so these are the fenestrated membranes together. So elastic fibers fuse together, creating this structure over here, network. So, and what's really happening is that smooth muscle cells that are in the tunica media contain a lot of the big rough endoplasmic reticulum. They contain a lot of endoplasmic reticulum, and they are producing reticular fibers and elastic fibers here. So smooth muscle cells they age. The smooth muscle cells in age produce more and more elastic fibers. And that's a big problem in one of the syndromes that you already know, Marfan syndrome. It's a big problem because there is a mutation in gene that's responsible for production of fibrillin. Fibrillin, as you already know from cytology, is a component of elastic fibers. Yeah, you know that from connective tissue, like the basic structure of elastic fibers is fibrillin. And if there is a defect in fibrillin, your body is not like it's not very how to say that. I'm lost right now. It's like you have very loose joints. Yeah, very loose joints and very long fingers, and also lens in the eye is connected via elastic fibers, and when they are not so elastic, they cannot put lens back to its normal position. Imagine it like rubber, or how to say this structure over here. When it's not elastic, it will become loose in a time. Yeah, so now it's very good, it's elastic, ouch, but in time it's not so elastic, and that's really what happens for the aneurysm of the aorta. Because imagine aorta with a pulse wave is going back to its own position, but when you have a defect of elastic fibers, it's not going to its own to its normal position. It's widening and widening in time, and aneurysm is other name for a dilation or the widening of aorta, and it's going to dilate and dilate more in time because there are no elastic fibers inside. And of course this process will end eventually, because aorta will rupture probably in some time. Yeah? So there is a rupture of the wall in aorta in these patients. That's very life-threatening situation. And this is the most dangerous thing that can happen. So, okay, you have some typical phase, loose joints, long fingers, that's okay. You probably cannot see very well, but you can cope with that. But you cannot live with the rupture of aneurysm of the aorta. That's very life-threatening. So that's why you need to see those patients regularly to control them. If the aorta isn't widening, so you need to provide some kind of operation to just treat the aneurysm. Yeah. Also, another type of syndrome, it's called Ehlers-Danlos syndrome, also hereditary problem. Also with the production of collagen and other stuff, so this might be a problem here. So you should know Marfan syndrome right now. Okay, so let's go for another type of artery. These are the middle-sized arteries, and these are no longer elastic types, so they don't contain those elastic membranes, but they do contain elastic fibers, but we are not able to find membranes, fenestrated membranes. So this is a muscle type artery, a small sized. What we can see here, this is the tunica intima over here, which is made of endothelial cells over here, and this layer right here is the tunica media made of smooth muscle cells mostly, but also containing elastic fibers and reticular fibers like normally. And next we have tunica adventitia, which is a thinner layer over here that is fusing or is connected to the loose connective tissue around. So sometimes you're not able to differentiate tunica adventitia because it's made of loose connective tissue, and also loose connective tissue is around the vessels, so sometimes it's hard to tell, but it is around, like right here around. And that's probably all I wanted to say. So here we have some diagram. So of course endothelial cells with basal lamina and some layers of smooth muscle cells here. What is important here is, as I said before, that artery of muscle type doesn't contain membranes, elastic membranes. But there is one thing that we can find in between the layers, and we can find an inner elastic membrane, one inner elastic membrane, which is curved. It's like right here, over here. This is a very special staining. This is probably a blue or green trichrome, but it's not important. But what we can see is a light wave over here. It's light because it's a blue trichrome, which stains connective tissue in different color. And this membrane, which is here, is between tunica intima and tunica media, and it's inner elastic membrane. So tunica media contains a lot of elastic fibers, and together in the border of tunica intima they create the membrane, the whole membrane. Also we can find the same membrane, which is a little bit thinner, in between tunica media and adventitia, but we cannot see it very well here. Don't worry, we're going to see it on another picture. So tunica media is a layer containing smooth muscle cells and is located between internal elastic membrane and external elastic membrane. And it's not like that aorta, which is an artery of elastic type, doesn't contain these membranes. We're not able to differentiate them in aorta because there are many other elastic membranes there, so we're not able to differentiate it. Okay, so this is how it looks like in our specimens. Sometimes you can see a nice layer, a wavy layer over here, and you should know from now on this is inner elastic membrane over here. It's also fenestrated, it's normal membrane over here. And also we have outer elastic membranes over here. It's hard to differentiate outer elastic membrane over here, but it's the last membrane that you can find over here. So this is the tunica media, and here we have tunica adventitia, and here there's a small layer of tunica intima over here, and there is a loose connective tissue all the way around. This is normal hematoxylin eosin staining over here. So this is very special staining here, and also this is special staining here, but here we can see no hematoxylin eosin, so that's why we can see the smooth muscle cells inside the tunica media over here. Very typical view of muscular artery. Next, we have arterioles, which are smaller cells. They are up to 500 micrometers, and they do contain about 1 to 5 layers of smooth muscle cells. That's very typical when you find a vessel with like 2-3 layers of muscle cells. Yeah, so that's probably arteriole. Okay, so these are the cross sections, and this is the longitudinal section of arteriole. So it looks like this, and these are smaller vessels. And now we have venules, and there are also many types of venules. We can see there are post-capillary, collecting, muscular, but you need to differentiate the basic venules that are here, that are about 200 micrometers to 1 millimeter wide. And here on this picture we can see post-capillary venule, which is a little bit smaller right here with erythrocytes inside. You can see it's special staining, it's blue, but don't worry about that. And we have nice adipocytes here around the venule over here, the post-capillary venule. These are the medium-sized veins. This is the medium-sized vein over here because this is the vein. And this is an artery over here. And this picture is very good because veins also contain normal tunics like arteries, as we described earlier in aorta or muscular type of artery. But there is a difference, as I told you before. Veins don't have so much layers of smooth muscle cells. So this is artery, of course, because we can see many layers of smooth muscle cells over here. This is tunica media, and there is of course also some tunica adventitia around here, but it's connecting with the loose connective tissue that's around, so we're not able to differentiate, but we can tell this vessel over here contains a lot of layers of smooth muscle cells, so this is an artery, of course. This is a vein, because what we can see are endothelial cells that are covering the vein inside, and it's very big, and it's not a circle like normal vessel. It's a little bit like I don't know how to say, like crushed together. We can see that it's some kind of disposition. This is very typical for the veins to not have normal position as a vessel like this in a circle. So sometimes they are even more dilated and curved a little bit, so this is very typical. And also tunica media, which is probably only over here, tunica media is very thin. Probably adventitia will be thicker, but once again we are not able to differentiate adventitia over here because everything is in loose connective tissue. So this is a typical picture, and this is how we can differentiate vein and artery. And they are not only 5 layers of smooth muscle cells, there are many of them, and that's why it's a medium-sized artery here, and also with the medium-sized vein over here. And these are the large veins. Oh, once again we have very special staining here, the green trichrome once again, but focus on this picture over here. This is a tunica intima, probably this layer over here is tunica intima, very thin layer. Then we have a tunica media, which is thinner than tunica adventitia, which is over here, tunica adventitia. And now what you can see, that tunica adventitia contains a lot of bundles of smooth muscle cells over here. And because tunica adventitia is oriented longitudinally, what we can see in tunica adventitia are these cross sections of smooth muscle cells, the bundles of smooth muscle cells over here. Tunica adventitia, once again, is the thickest layer of all tunics in large veins. Yeah, it's the thickest layer, and it contains a lot of smooth muscle cells inside, which are oriented longitudinally. So this is of course a cross section of a vein, because there is a cross section of smooth muscle cells. We have a slide that we are going to discuss during our practical training. We have a cross section of a vein and longitudinal section of a vein. And based on the orientation of layers, you should be able to differentiate, okay, this is a cross section, okay, this is longitudinal section. So that's why I'm telling you right now, this is very important, also during exam. And there is some adipose tissue around, that's pretty normal. Around veins, there's a lot of adipose tissue, so don't worry about that. So, and now let's skip to the heart. Let's skip to the heart, because heart also contains many layers. Not so many, but you should be able to differentiate 3 basic layers that can be found in the heart, and that is an endocardium. I'm sorry, this shouldn't be here. That's endocardium, myocardium, and epicardium. These are the basic layers. Also, whole heart is in some kind of cavity that is called pericardial cavity, that contains 50 to 70 milliliters of fluid. You should remember this number. What I wanted to say is that the pericardial cavity contains 2 layers. It's made of 2 layers, and it's made of visceral layer, and parietal layer. And visceral layer is really epicardium that can be found on the heart. And the pericardium is parietal layer that is more away from the heart, and between these 2 layers between them there is a fluid. The mesothelium is a special epithelium, is a single layer flat or cuboidal epithelium on the surface, and is covering also epicardium and also pericardium. So when we have some kind of cavity, there needs to be some covering of the cavity. Yeah, so mesothelium is normal epithelium that can be found also in abdominal wall, in pleural cavity, and also in the pericardial cavity. So remember this name right now, mesothelium, right here. This is a histological picture of the heart. Here is here is atrium. This is the ventricle, so we can see the wall of atrium, the wall of ventricle. Now you can tell some differences. We have endocardium over here. That's the first layer that lines the atria and also the ventricles. Is a little bit thicker in atrium, so thicker endocardium in atrium, thinner in ventricles. Endocardium has many layers. We're going to discuss them. And under endocardium we have myocardium over here, which is composed mostly of cardiomyocytes, and of course ventricles contain thicker myocardium over here. Atrium contains thinner myocardium, so that's the difference between these two. And also we can see epicardium over here, which is a, as you know, visceral layer of a cavity right here. So there is fluid around the heart. It's normal to have fluid around the heart because it prevents the friction. It's for the sliding because heart is moving a lot, so there need to be some sliding. And what we can see also in this picture, we can see a valve over here, which is made of endocardium mostly. It's a duplicator, so two layers of endocardium that are fused together are creating a valve. So endocardium is made of some layers. Here we have endocardium, which is in the atrium because it's thicker than endocardium, which is in the ventricle. Here is myocardium, so it's still here, but you can see it probably. It contains layers which you cannot differentiate very well. You can differentiate them in atrium better because it's bigger than in ventricles, but you should know the basic layers of endocardium. So first we have endothelium, of course, that's on the surface, which lines the lumen of the heart. So here we have blood, of course. Then we have some subendothelial layer over here, subendothelial layer, which is a very thin layer of loose connective tissue under endothelium. Then we have elastic and muscular layer, so elastic muscular layer, because what can I see here are smooth muscle cells over here. So elastic muscular layer. And here under elastic muscular layer, here we have subendocardial layer over here, subendocardial layer, which is once again made of loose connective tissue. It also contains a conducting system, heart conducting system, and it's called subendocardial layer, but it's part of endocardium. Okay, so we have 4 layers over here. Okay, this can happen. Endocarditis is the inflammation of endocardium, and it sometimes happens that bacteria will sit in the valves. So here we have an atrium right here, and probably down here will be the ventricle. Here is the ventricle, and we have valves here, which are like duplicators of or duplication of endocardium itself. And acute endocarditis happens mainly on the valves. So we can sometimes find these vegetations that are the microthrombi, the thrombi with bacteria sitting on the walls, and it's a very dangerous situation that needs to be treated perfectly with antibiotics and stuff like this. So it's very important for you to know right now what's endocarditis and where we can find endocarditis. So, and also I mentioned the conducting system. And the conducting system is a very special system that is composed of many structures. We have sinoatrial node close to the vena cava superior right here. So sinoatrial node is made of very special cells, very special cardiomyocytes, and they are contracting by themselves. Also, they are innervated by parasympathetic and sympathetic nerves, but also heart can contract by itself. Yeah, heart doesn't need any nerve connection from the nervous system. It can contract by itself, but you need parasympathetic and sympathetic nerves to modulate the contraction of the heart. Okay, so we have sinoatrial node, which is connected to atrioventricular node, which goes to the bundle of His, and then it's divided into left and right branch, which are then divided into smaller branches, which are called Purkinje fibers. Purkinje fibers. Over here, you're going to learn more about this system probably in anatomy and in physiology, but right now remember Purkinje fibers as the last step in conducting system, and these Purkinje fibers as the whole conducting system are made of modified cardiomyocytes. What we can see here is a picture you probably know from electron microscope. Here you can see myofibrils, and this structure over here is intercalated disc right here. So each cardiomyocytes are connected with intercalated disc. So this is a cardiomyocyte, and this is the second one over here. We can see myofibrils. You should know what structures can be found in intercalated discs. Okay, so and if you don't remember, please go back to the previous lecture from a muscle tissue, or just read some book or whatever. You should know the three basic components of intercalated disc, like the fascia, adherens, the desmosomes, and the gap junctions that are there. Yes, it's written here. So, and the epicardium. So here we have a nice myocardium. We have bundles of cardiomyocytes over here, and epicardium is a layer of loose connective tissue that contains also some nerves, maybe some small vessels, because coronary vessels that supply heart, coronary vessels, are located in the epicardium. It's like tunica adventitia. Okay, epicardium is something similar to tunica adventitia because it also contains vessels, and you can find adipocytes over there. And what we can see over here is that epicardium is covered with mesothelium on the surface. And mesothelium is epithelium, normal epithelium, which is flat, it's single layer of flat cells or slightly cuboidal, and this is normal finding over here. So, and now just a little bit about atherosclerosis right here. So really, atherosclerosis, you should know something about it because it's a disease that's affecting most of the population. It's in time, like every person will develop some kind of plaque inside the vessels because of the cholesterol, the hypertension, and other risk factors that are there. It's an inflammatory process. Now we know it's inflammatory process because normally you have normal endothelial cells that line vessels, but there is some kind of lesion to these endothelial cells because of the high pressure, or there are many cholesterol particles inside, and they start to go to the intima, tunica intima. So they start to go under endothelial cells, these cholesterol molecules, or as I mentioned earlier, LDL particles. And they start to accumulate in tunica intima and form a plaque. This disease is developing in years, of course. It's not just like, okay, now you have a plaque, but it's developing in years. And you can see like what it means, like the sclerosis mean like hard, it's hardening over time, and a tear is a mesh dough or some kind of paste which is made of these lipids, the cholesterol, the foam cells. I'm going to explain. You can see right here, these are the fat containing macrophages over here, and it's an inflammatory process, so remember that. It's causing degeneration of the walls of arteries, and it's a dangerous disease, very dangerous disease, because there are many causes of atherosclerosis, like the obesity, the smoking, hypertension, diabetes, and there are also hereditary cases, and it's starting at males 10 times more than at females. We can find atherosclerosis 10 times more than in females. And what I wanted to explain is a pathogenesis, maybe a little bit, because you're going to talk about it in pathology. But right now you should know that's probably starting forming also in our bodies right now, because in your bodies also, because you're young, but you probably have some small plaques already inside your vessels. And what's happening right now, so imagine this endothelial layer of cells, there is some kind of lesion to these endothelial cells, and what's happening is that LDL particles are going inside here. So here we have LDL particles inside. And these LDL particles are oxidized because their normal cells are releasing reactive oxygen species. So they are oxidized, these LDL particles, and they release cytokines. Cytokines are just small substances that can tell inflammatory cells that come and get me, I'm releasing cytokines and come and get me. Yes, so inflammatory cells then go inside also this tunica intima. For example, monocytes go inside, and as you know, monocytes will transform into macrophages. And these macrophages will, of course, what are they going to do? They want to clean the vessel, so they're going to eat LDL particles here in the tunica intima. But these oxidized LDL particles are different in that macrophages cannot recognize if they are full enough, so they're going to eat till death. Yeah, so these oxidized LDL particles are very difficult for macrophages to digest, because they cannot tell, okay, now I'm full, it's enough for me. So what are they going to do? They're going to die. And first they turn themselves into something called foam cells. So foam cells are just macrophages that eaten up many LDL particles. So they are filled with lipids inside them. So that's why they are called foam cell, and eventually they are going to die. So the plaque of atherosclerotic plaque is the macrophages that eat up a lot of LDL particles and die eventually there. And okay, so the artery or the vessel is going to try to reparate somehow. It's try to fix this problem. And that's why we have smooth muscle cells over here. The smooth muscle cells start to migrate over here. They start to migrate, so they can move a little bit, start to migrate, and they start to produce of collagen and elastin over here, and they will create a fibrous cap around this plaque because they want to hide it, they want to cover it. And so that's why they're going to produce fibers of collagen and elastin around. They want to cover it because probably there is some kind of disruption of endothelium, and underneath endothelium there are cells filled of lipids, and this is very dangerous because sometimes there are many lipids under this fibrous cap that is now around. Sometimes this fibrous cap can rupture. Normally it can rupture, and these lipids that are under this fibrous cap can be released to the bloodstream. And when you have free lipids in the bloodstream, when you don't have some kind of particle like LDL, VDL, VLDL or stuff like this, when you have only free lipids inside, they are very thrombogenic. They are thrombogenic. So the thrombus will form here because erythrocytes, which I'm trying to draw, erythrocytes start to stick to the lipids over here, and they form a thrombus. So, and this thrombus is of course very dangerous for the vessel because it can occlude the vessel and stop the supply of blood into the tissue, and this is happening in the heart. So in myocardial infarction, what's really happening, these are the coronary vessels which are filled with these macrophages filled of lipids because of LDL particles that are inside. There are many of them, so they are going inside the vessel, the wall of vessel. There are many inflammatory cells inside, so there is inflammation going on. Smooth muscle cells try to cover this whole process, but they are insufficient enough sometimes, so the plaque will rupture. It will release the lipids that are in the tunica intima, and the thrombus will form, and from the thrombus that is here we can create embolus. So this is the thrombus, but sometimes it can rupture, everything can rupture, and the thrombus can move with the blood flow, and when it moves with the blood flow it's called embolus. So we probably think right now, okay, so this is more of pathology, but also Dr. Becke was explaining this process to Czech students, so I want to stress it also for you because it's very important, and it's one of the most important pathophysiological processes ever, and you're going to hear more about it, and I hope you understood everything. Also, it's not going to be in oral exam or in tests, so don't worry, it's just for you right now. We can see those plaques also in the walls. So this is normal tunica intima over here, but now you can see it's thickened. This very thickened tunica intima is not normal for tunica intima to look like this. This is a fibrous plaque because also we have a fibrous cap that is covering the plaque, but there is fibrous tissue forming also here, many fibers, collagen, elastic fibers inside tunica intima. This is not normal. In this picture we can see foam cells, so you can see they kind of look like multivascular adipocytes, which are in brown tissue, brown adipose tissue, but these are normal macrophages that are filled with lipids. So these are foam cells over here, foam cells. So you're going to hear about this more in pathology. And this is how it looks like macroscopically, grossly. This is atherosclerosis of the abdominal aorta, which is pretty thick aorta. This is a bifurcation. It's dividing into common iliac arteries, and so you can see how it can looks like. It's very, very dangerous. So thank you, and goodbye, and see you next time.